I don't know. I'm not hearing that properly from you.
Shall we wait for 2-3 minutes, madam, for them to join? I'm asking you for a request.
Okay.
Good afternoon, ma'am. This is Shweta.
Good afternoon, Shweta. Good afternoon. Good afternoon, ma. Tehera. Only you three girls are regular in the class. I don't know what about the rest of the class. Never to be seen. Anyways, we'll start with our class. So we have
Ma'am, I have a doubt from yesterday's class.
Pardon, ma'am?
I have a doubt from yesterday's class, ma'am.
Yeah, go on.
Yesterday we have seen the gene transfer via cells. Okay, in that we have seen generation of chimeric mice and generation of the cloning, that is sheep. Two methods. Okay, pluripotent cell and differentiated cell.
Yeah.
So here we are transferring when we are using pluripotent cells, we are transferring the blastomere. So to the blastocyst. So doesn't change the genetic component, ma'am?
It does not. What happens is when it is a hybrid, then you can change the genetic component. Here, it is only expressing the genes that have been transferred as a chimeric form. Chimera is something like incomplete recombination.
Okay.
So you want to know until you get a complete homogeneous characters, you will have to keep on backcrossing the chimeric form until you get a homozygous cells that is present over there. So yeah, you want to change the genome, but not at once. It's not going to be at once. You will have to keep backcrossing the product that you got, the transgenic founder that you got. You have to keep backcrossing it until you get a homozygous genes that is of your interest. Only then they will be expressed properly in the progeny.
No, differentiated cell, when we are using differentiated cell, there we are removing the nucleus from the oocytes. But here we are not removing the nucleus.
We are not removing. That is the reason why you get a chimeric form over there. But in an undifferentiated form, you are adding, you are removing the nucleus from the egg, and you are adding the fibroblast.
Yes.
So when you are adding the fibroblast, you are getting a clone of that animal which you have added, the nucleus of that animal which you have added.
Yes, nucleus contains chromosomes and genes, no ma'am?
Exactly.
So we are adding the new genes to it so that we can get the clone. But in pluripotent cells, we are not removing the nucleus and we are adding more genes.
More genes.
Does it occurs any genetic changes in the organism?
That is the reason why you want them to do the pluripotency, right? So you want to have a change in them, but the change is not at once. The change is not at once. You have to do multiple crosses. The picture that we show, it's just directly you add it and you're going to get a product. It's not like that. Once you get the product, if you can check it on the pluripotent stem cells that we did yesterday, I told you all that there's a chimeric organism that is being formed. Remember the chimera, the black and white colors that you get?
Yes. Yes.
Hello? Yeah?
Hello. Ma'am. Yes, ma'am. I remember.
Yeah, you remember that. So that's a chimera wherein the genes have not been completely incorporated into the genome. That is, it is not homozygous characters not to be seen. So when this chimeric gene or chimeric animal is going to be backcrossed with a wild type, you're going to get a heterozygous one first. That is complete black color mice we saw after that. Now this complete black color one is heterozygous for that particular genome which you had added. That is one chromosome is going to be different from the other. So that is why you're calling it as heterozygous. One chromosome has got the genome that you have transgene, and the other chromosome is going to be a wild type. So that is the reason why you call it as heterozygous. Now, when this heterozygous is again cross with another heterozygous, you have one dominant and one recessive. Until you get both the chromosome recessive, you will not get a homozygous characters. So when you get a homozygous genes, that means you have transformed that gene of interest into the organism. Until then, you'll have to keep crossing the organism after adding the transgene into the genome that is in the blastocyst. It's not a one step. It's it has to be a multiple step over there.
Okay.
Understood?
Thank you, ma'am. Yes, ma'am.
Still, if you have any doubt, we will talk about that after the class. Okay.
Oh,
yeah.
Okay.
Okay. So we'll start with the class now. So today we will talk about aquatic biotechnology. We have completed block 1, completely block 1 we have completed. Now we'll talk about block 2, that is applied biotechnology. So we are going to talk about applied biotechnology. In that, I wanted to talk about aquatic biotechnology because this is of even more greater importance for zoological point of view. So I thought I will complete this aquatic biotechnology first. So coming to the first topic over there, under aquatic biotechnology, what you're going to do is, what exactly we are going to study in this aquatic biotechnology is, we are going to, we must have heard about green revolution, we must have heard about the white revolution, and I hope you must have also heard about what is blue revolution, right? So when you're talking about a blue revolution, the blue revolution is nothing but we are going to take care of or increase the production of fishes in the water. So production of fishes, increasing in the number of fishes and taking care of them, and biotechnological point of view, what you're going to do is all what you call it as blue revolution. Okay. So when you're talking about this blue revolution, why we are going to give so much of importance to the fishes is because first thing is when we were talking about aquaculture in a class, I told you all that fishes are going to have lot of protein content in them, and they are also going to have less amount of cholesterol present in them. So both protein content is more and less amount of cholesterol means definitely this is a much accepted version of meat when you talk about or when you compare with the chicken or a mutton pieces over there. So this most of the population are switching from meat eating to fish eating generation. So government is also trying to take steps so that they can increase the production of aquaculture, increase the production of fishes, and control its disease present in them, so that you can supply more amount of fishes to the public. So aquaculture or aquatic biotechnology is a budding subject wherein there is a lot of work going on in that. So here we're going to, till now we talked about transgenic mice, transgenic rat, transgenic cows and sheep. Now we are going to talk about transgenic fish, how it is going to be important and how it is going to help the human mankind. So when you're looking into this picture, you can see there are two types of fishes that I kept over here. So that's salmon fish. Usually you find it in Atlantic oceans over there. In the Western countries, mostly you'll find this salmon fish. And then you have this medaka fish over there. That is how a medaka fish looks like. Mostly you find it in the Japanese country over there. Most of them is present over there. So this is also called as white color because white fish. Oriza fish, the other name for this medaka fish is a Oriza fish. Why it is important? When you're talking about transgenic fishes, when you genetically engineer this transgenic salmon for more rapid growth using growth hormone gene under the control of the ocean pout antifreeze protein gene promoter at 3' untranslated region. So what do you mean by ocean pout antifreeze protein gene? Now, when you're talking about the fishes, when you're talking about the Atlantic oceans, you will definitely find that there is always the water at a freezing level. So the water is always at freezing point over there. So you can expect the fishes also to freeze and die. But that doesn't happen in some fishes, wherein they have this anti-freeze protein. So whenever the water is getting colder and colder, wherein the water is going to get freezing point, that is when they form ice, there is this protein present in the fishes, which is produced at that moment. They hug around the ice or the freezing water that is being formed, thereby controlling the freezing of the fishes. So that's called an anti-freeze protein. And that's the reason why these fishes do not die in the Atlantic Ocean or any other places where there is, when the water is going to be very cold. So this salmon was genetically engineered, where they found this anti-freeze protein, that is a promoter region, and they added these growth factors. And with what they found is, this is the normal size of an salmon fish in the water when it's going to grow normally in the water. But when you transgene these fishes, again, this is a point over there. Transgenic fishes is transgenes where you want to add that particular gene of interest so that it gets incorporated in its genome and that is expressed. But this expression is not a one-step express. You have to do a lot of steps wherein you have to add the gene of your interest in the vector, and then that vector goes into the blastocyst. Then they have to be expressed in the progeny. Either it can be a F1 progeny or an F2 progeny or an F3 progeny. It's a continuous process over there. It's not a one-step process. And in that process, you might have so many fishes dying or so many fishes not expressing. So it's a tough process, but nevertheless, when you get it, then you're going to really be happy with the result. So this is the result of the salmon fish, which this is a transgenic fish where the human growth hormones was added to it. Yesterday, we saw a mice, which was a super mouse, which was really big in size when you compare it to the parent. The same thing over here. So this is the normal fish, which grows to this very small size, whereas this is a large fish, which is also called as a transgenic fish, wherein you have introduced human growth hormone, where you are adding this because it should have a promoter region over there. So if you can see in this picture over here, so this is the promoter region, and this promoter region, you have to find out where it is present, and then you can add your gene of interest. Only then that will be expressed. If it is going to be inserted in any part of the genome, then that gene is not going to get expressed because it is not having a promoter to promote the expression of that particular gene. So promoter gene is most important in when you want to do any transgenic fishes. So here you need a promoter gene. What is a promoter gene? This is an anti-freeze protein promoter gene that is have to present. That is a AFP gene. Okay. So there's one more fish, which is called as Medaka fish, which was genetically engineered to serve as biosensors for environmental pollutants. So biosensors for environmental pollutants, because there's a lot of pollution that is occurring around in the sea and all other places, because all the oceans and the seas are being contaminated. So when these fishes have to survive in those contaminated water, what happens is these are not able to survive in those particular environment, and they usually die over there. So you're preparing this fishes so that whenever there is pollution in the water, the fish starts to glow. So you can see over here the glowing kind of proteins gene that is present. GFP is glow proteins, which will glow in the water whenever there is pollution in the water. So this is done by the estrogens over there, by using an estrogen inducible promoter region over there. Now, what is the specific function of estrogen? It is a vitellogenin promoter. Remember, when we were talking about a vitellogenin in the crab, we were talking about the yolk formation. Vitellogenin promoter is for the yolk formation. So this estrogen gene is needed as a promoter gene so that it can incorporate this glow protein that has to be present. And whenever there is pollution, the fishes will start to glow. That is an indication for us that the water is contaminated, and we have to treat the water so that it can become fresh again. So that is one more point over there. And these fishes are also been genetically modified to secrete various human hormones, and they express promoter sequences from other fish. And this is also made to make antimicrobial proteins. So these are various ways where you can use this fishes for transgenic purposes. So this is how Medaka fish looks like in the Japanese oceans over there. Okay, so introduction to the transgenic fishes. The transgenic fish was first produced in China in 1985. Why in China? Because China is the first country when you're going to talk about a production of aquaculture. This stands first in the world when you're going to talk about aquaculture production. So as of 2013, approximately 50 species of the fishes have been subject to genetic modification, and the resultant is more than 400 fish traits you can find in combinations. And this has been modified for most of the food species fishes, that is wherein people love to eat this type of fishes all over the world. That is Atlantic salmon, tilapia, and common carp. So this is an Atlantic salmon. You can see how big the salmon is. Then this is tilapia, and this is European carp, or also called as cyprinus carp. Now, how to differentiate between a tilapia and a common carp? You can see the dorsal fin over there. If you can see the dorsal fin over there, the dorsal fin over there starts at almost near the operculum. So if you can see the operculum over there, from there, the dorsal fin starts over there and ends up in a ribbon-like shape. Whereas in a European carp, you can find the fin, the dorsal fin starting almost near the thoracic region, just beginning of the abdomen and the thoracic region, in the mid of the fish body, you will find this dorsal fin occurring. And also they have those whiskers that are present on the lower jaws over there. So this is how you are going to identify or differentiate between a tilapia and a European carp. And a European carp also has, if you can see the caudal fin over there, the caudal fin is bifurcated or fork-shaped one, that is you find. But in tilapia, you have a almost square or square-shaped truncated caudal fin that is present. So this is how you're going to differentiate between species of the fishes over there. So these fishes are usually the most consumed fishes, and that is the reason why transgenesis was done in these fishes, so that it can grow faster and production rate is much higher, and then you can produce more amount of fishes in a given short period of time. So the genetic engineering of transgenic organisms differ from gene delivery because the former involves integration of exogenous DNA. That is, transgenic organism means you are involving or integration of exogenous. That is, you like one gene of interest, that is, for example, a human growth hormone. You're going to take out that human growth hormone and you're going to introduce it into the host genomic DNA. So thereby you're modifying those gametes over there. Whereas in the later, it is simply the insertion of a gene into a cell. Insertion of gene into a cell is a different type, wherein you're going to only deliver that particular gene of interest over there. So that is how you're going to differentiate between transgenic and or just involving introducing a gene of interest in a cell over there. So interestingly, fish offer easier genetic manipulation compared to mammalian counterpart, because what it means is, yesterday in the earlier classes, we saw the transgenic cow, mice, and sheep over there. Their manipulation is a little bit difficult, but in fishes, it is easy. Why? Because the fishes are going to lay many number of eggs. That is almost, if you're going to talk about katla, 1 kg egg can give up to 2 lakhs eggs. So this large number of eggs that can be laid by the female, one thing. And the first report of the generation of transgenic fish in the aquaculture was found in goldfish. So the transgenesis were done in the goldfish and rainbow trout. And over a period, a growth hormone has been targeted as a transgene, aiming to increase the growth rate in more than 20 teleost species. So the teleost is freshwater species is present, and this has been done in this fishes over there. Now you can see in this fish over there, this is what you saw in the earlier picture over there. So this is the normal growth of the fishes, but when you add human growth hormone into them, you can find within 12 months, this is the size that you can have. So that is a tremendous size that you can get in a very short period of time. So that is the importance of transgenic fishes over here. Okay. So what can transgenic technology offer when you're talking about aquatic biotechnology? So you have 6 factors, important factors. Complete control of reproductive cycle of the fish species in the culture, excellent genetic background of the brood stock, efficient prevention and detection of disease infections, and through a thorough understanding of the optimal physiological, environment, and nutritional conditions for growth and development. So if you're understanding what is the type of physiological characters that it needs for the fish, the environmental conditions, whether it is a fresh water, whether it is a salt water, or whether it is a brackish water, and what is the type of food that you have to give them for the growth, all this can offer a better technology for that transgenic technology. Sufficient supply of excellent quality of water, because water quality should be most important. Some freshwater fishes, they survive in a pH 6.8 to 7.5 type of pH water, whereas in the salt water, the pH is a little more higher. So application of innovative management techniques can also be helpful in transgenic technology. So by improving these factors, aquaculture industry has developed a remarkable extent during last decade. And to sustain this growth, application of various newly developed technology in molecular biology, including transgenesis, have proven worth in the modern aquaculture industries. So this can also employed for enhance of the growth rate, control reproductive cycles, and improve feed compositions. And also you can produce new vaccines and develop disease-resistant genetic stock. So in your book, they have given types of transgenes. So you're going to talk about 3 types of transgenes over there. What are the 3 main types of transgene? Transgene constructs designed to add new function. So you want to add a new function to that particular genome of interest to get the transgenic individuals. So for example, over here, when you're going to add a new function, you're going to add growth hormone transgene so that you can show a better growth, better enhancement of growth in that particular fish. The second one is transgene constructs generated to assess the strength of the promoter enhancer element in question. That is, you want to see what is the strength of that promoter enhancer. So for example, you are going to fuse a particular gene, fusion of the structural gene of beta galactosidase or luciferase to promoter enhancer elements whose strength is observed and measured. So you want to know, when you're inserting a gene of your interest, what is the strength of that promoter enhancer, how well that promoter enhancer can do. That is, here we are going to talk about beta galactosidase as a enhancer over here. So how good this enhancer is to enhance that particular gene which they have to show it for enhancement of that particular character. Then the third one is transgene constructed to interfere with the expression of specific host gene. So you're going to construct a gene of interest wherein you want to suppress some particular expression in that wild type of fish. So you're going to add some kind of a gene so that this goes and joins the binds to the genome so that this can suppress the expression of that particular. So for example, when you're talking about P53 is a tumor suppressor gene. But when you wanted to add a mutated P53, you want to mutate the expression of tumor suppressor gene so that the animal can express cancer genes over there. So this is what is interference with the expression. And this interference with the expression is done by antisense RNA. So when this antisense RNA is introduced into the genes, this is going to interfere with the expression and it's going to mute the expression, whereby that particular expression is not expressed in the wild type. So these are 3 types of transgenes that we will be talking about. Okay, so method of transgene fish production. So how we are going to do this transgenic fish production methods over there. We talked about 3 types. Now we are going to talk about methods, how we are going to construct this transgene in the fishes. So depending on the purpose of the transgene studies, you're going to talk about 3 main types. That is again, gain of function, report of function, and loss of function. So you're going to talk about gain of function, report of function, and loss of function is what you're going to talk about over here. So what is the point over here? When you're talking about this gene structures, you need to have an enhancer over there, the enhancer, and then you need a control elements, and then you need a promoter gene over there. Promoter gene is also called as Tata box. Why Tata box? That is the gene that is present over here, TATA. Usually it is called as Tata box. That is a promoter region. And from the promoter region, when you add this gene of your interest, that is, you have three gene of interest, that is open reading frame over here. Which are the open reading frame over here? Exon 1, exon 2, and exon 3. So these are the exon 1, exon 2, and exon 3. These are the open reading frames. Introns are the ones which does not help in expressing the genes. So they are very, very secretive and very much highly coiled over there. And this is a downstream regulatory where it is going to terminate the expression of that particular protein of interest over there. So what is this gain of function? The transgenes are designed to add new functions to the transgenic individuals or facilitate the identification of transgenic individuals. And if the genes are expressed properly, then it is called as transgenic individual. If it is not going to express, then the experiment is failed over there. So transgene containing the structural genes of fish and mammalian growth hormones are fused to the promoter, which led to the greater production of growth hormones. So you have the promoter region over here. Beta actin gene is a promoter over here in this fish. So the beta actin gene promoter, proximal promoter region over here. And wherever you have this arrows pointing upwards, this is where the gene expression is being enhanced. And wherever you have an arrow pointing towards downside, the gene expression is being silenced over there. So this is where when you add your growth hormones, the gene expression is enhanced or it is expressed over there. So that is the reason why it is very much useful to check where the promoter region is. So when we are reading and studying over here, it is so easy to understand. You have a promoter region, you have to add your gene of interest over there, and then you have to have a silencer gene or a nonsense gene over there, where the stopping of expression is occurring over there. But when you're going to talk about this in practicality, then the work comes over there where you have to search where the promoter gene is, how you're going to find out an endonuclease, where you have to restriction endonuclease, where you have to cut it, and then you have to add your gene of interest, and then you're going to ligate. It's a pretty tough experiment, but this is what you have to understand when you're going to talk about it in theoretical point of view. So this is gain of function, where you're going to add your gene of interest beside the promoter so that it is going to be expressed. That is called as gain of function. Then the next one, what you're going to talk about is the reporter function over here. So what is a reporter function? The reporter function transgenes are commonly used to identify the success of gene transfer effort. So how successful you were in transferring that particular gene of your interest. So this is done by bacterial chloramphenicol acetyl transferase, that is CAT, and you also have beta galactosidase or luciferase genes fused to the functional promoters. These are the example of reporter function transgene. Following gene transfer, the expression of the reporter gene activity is used to determine the transcriptional regulatory sequence of a gene or the strength of the promoter. So this is a reporter gene which is going to report to you that how successful you were in transferring that particular gene, and whether the promoter genes are helping them to express that particular gene of interest that you have added. That is reporter function. Now, loss of function. Now, what is loss of function? The loss of function transgenes are constructed for interfering with the host gene. That is, when a wild type is there, wherein the gene is being expressing thoroughly, what you're going to do is you're going to add a catalytic RNA or any antisense RNA that cleaves the specific mRNA and thereby cancel the production of normal gene products. So that is loss of function. So when you want to stop that particular gene to express, you're going to add something which is going to interfere in its expression, and then they are completely, once they are going to get transferred into the genome, this particular antisense RNA is going to stop the expression of that particular gene. So that is called loss of function of that particular expression over there. So that was about gain of loss, reporter gene, and loss of gene functions over there. The next one is selection of fish species. Now you cannot do this type of transgenic in all the fishes. You have to identify a particular fish in which you want to work, whether it is good enough to be done or whether you cannot do it. All this has to be taken care of. So for this, what they found is some catfishes, common carp, goldfish, Japanese medaka, that is what we are talking about in the earlier class, medaka, loaches, northern pike, salmon, rainbow trout, tilapia, and zebra fishes were found to be best for transgenic transfers over there. So here you can see zebra fish that has been kept. These are very small fishes, very easy to handle them. And also they they release a lot of eggs. So you have lots of eggs to conduct this experiment. It is not 1 or 2 or 3 eggs over there. So you have to be very careful. And why are these zebra fishes or medaka fishes or this type of fishes is selected? Because it is having a short life cycle. That is, they can survive only for 3 months from hatching to mature adult. And the mature female is going to produce hundreds of eggs on regular basis without exhibiting a seasonal breeding cycle. So you don't have to wait for a seasonal breeding cycle. So yes, something like when you're talking about telios over there, when you're talking about rohu or katla, and this rohu, katla and all, they're going to breed only in the month of May to July, but otherwise they're not going to release any eggs over there. Why zebrafish? Zebrafish, because it is having a short life, because they're very small ones. They have a very short life. They can produce hundreds of eggs, and you have enough eggs to work on this gene transfer. And this can also be maintained in the laboratory for 2 or 3 years in a cycle. That is, the eggs can be freezed and kept in the laboratory for 2 or 3 years. So eggs from these two fishes are relatively large. What is the diameter? 0.7 to 1.5 mm diameter. You can see the eggs over there. And they possess very thin semi-transparent chorion. So they have a very thin transparent chorion around, and it's easy for micro injection of DNA into the eggs. So you can easily inject anything that you want into this pronucleus of your interest. Besides, availability of the inbreed lines and various morphological mutants of both these fishes have made them most suitable organism for conducting gene transfer experiment. You can see over here very clearly how the eggs, they are so transparent that you can see the growth with inside the how well they are going to grow, because they are transparent over there. Okay, so method of gene transfer. What is the methods of gene transfer that you're going to do in this particular fishes? Various techniques such as calcium phosphate precipitate, direct micro injections, lipofections, retroviral infection, we are going to use virus over here. Electroporation, you're going to introduce electric shock to them. So that's called as electroporation. Remember, I was talking about this electroporation in the yesterday's class also. This is done by particle gun bombardment and has been used to introduce foreign DNA into the animal cells or plant cells or germline, mammals and other vertebrates. So first, under gene transfer, we'll talk about micro-injection of eggs and the embryo. So micro-injection of foreign DNA into newly fertilized eggs. So this is a newly fertilized eggs, and you're going to introduce this transgene into this newly fertilized eggs, and has been adopted for introducing transgene into Atlantic salmon, common carp, goldfish, rainbow trout, tilapia, and zebra fishes. The gene construct that were used in these studies include human or rat growth hormone. What is this gene over here? This can be either a human or a rat growth hormone. And rainbow trout and salmon growth hormones, cDNA, chicken delta crystalline gene protein, winter flounder antifreeze protein, antifreeze. Remember what is an antifreeze over here? E. coli beta galactosidase gene and E. coli hygromycin resistant gene. So these are the different types of transgenes that are being used to introduce in this particular fertilized eggs. Once this transgene has been introduced, this is again incorporated into the pregnant or a female fish, and then ultimately it's going to give different types of fishes because they are going to lay too many number of eggs. So among them, only 1 or 2 might going to have this transgene added into this genome, as I told you all. So not all fishes are going to incorporate this. We don't know which blastocyst is going to take up this gene, incorporate it into DNA, and you have to go and find out them. How you are going to go and find out them? So once this is being produced, this particular gene is called as a founder fish. This founder fish is again going to be backcrossed, and then you're going to do it until all the progeny is going to be of that particular transgene. That particular transgene as homozygous transfer of that particular gene. Until then, you're going to keep searching for it by back cross, test cross, and so on and so forth. So yeah, the steps for micro injections. What is the steps that you need for micro injection? Eggs and the sperms are collected in a separate dry containers. So once, because fishes have external fertilization, so you're going to collect the eggs and the sperms. You're going to collect in a dry container, and fertilization is initiated by adding water and the sperm and eggs with gentle stirring. The injection apparatus consists of a dissecting stereo microscope. So this is a dissecting stereo microscope that you will have, and two micro manipulators, one with glass micro needle for delivering the transgene. So you have the micro needle. So here this is the micro needle over here to deliver the transgenes, and other with a micro pipette holding the embryo in place. So other one is a micro pipette that is going to hold the eggs in place so that you can inject the genes, transgene that you want to transfer them. Okay, fertilized eggs are then micro injected with first 2 hours of fertilization. So within first 2 hours of fertilization, you're going to do this micro injection. Routinely, about 106 to 108 molecules of linearized transgenes with or without plasmid DNA is injected into the egg cytoplasm. Depending upon the fish species, the survival rate of the embryo ranges from 35 to 80%, while the rate of the DNA integration ranges from 10 to 70% in the survival. So not all fishes are going to survive, and not all fishes are going to exhibit this transgene in them. So tough chorion of the fertilized egg of some fishes like rainbow trout and Atlantic salmon can frequently make insertion of these glass needle difficult. So because they are having a tough chorion, it is not as transparent as in the zebra fish or medaka fish over there. So these difficulties can be overcome by one or the other following, that is inserting the injection needle through micropile, making an opening in the egg chorion by microsurgery. So you have to just make a small surgery over there so that you make a hole and then you can insert the gene of your interest. Removing the chorion by mechanical or enzymatical means. Enzymatical means is you can also use trypsins over here to enzymatically remove it. Reducing chorion hardening by initiating fertilization in a solution containing 1 mm glutathione. What you're going to do is over here. So when you're going to add this eggs and the sperms, and by gentle stirring, when you're going to add water, instead of water, you can also add this solution containing 1 mm glutathione. So what this is going to do is, this is going to gently loosen up the chorion over there. So it reduces the hardness in the chorion. So it is easy for you all to inject the gene of your interest. And injecting the unfertilized egg directly. So you can also do all these steps over there for micro injection. So what you're going to do is, this is what we are talking about. You're going to collect the egg and the sperm, and you're going to fertilize them. And after fertilization, you're going to add this exogenous DNA of interest by micro injection. And after that, you're going to introduce that in a female fish. And then ultimately what you get is a mosaic. So this is a chimeric fish that you're going to get, transgenic chimeric fish, because not all fishes are going to have it. And those that have had it, they're going to have a combination of these two characters present in them. Then once you are going to backcross it with a wild fish. So this is a wild fish over there. When you're going to backcross it, then you're going to have a progeny, which is going to be either non-transgenical animal, or you're going to have a transgenical animal of the interest. So this step that we are showing over here is not one step directly. You have to do multiple times to get this particular transgenic animal of your interest. So it's not a one-step one, which you're going to see in this particular picture. So this is a microscope that you'll be using, a stereo microscope. It has a ultra precision straight forceps over there. Then you're going to have a plastic hand rest where you can rest your hand and do the experiment. Then you have a 3-dimensional micro manipulators, and you have a manual micro injector. So how you are going to inject the genes of your interest, and a simple magnetic stand is present over here. So these are the setup where you're going to do, keep everything ready when you're going to do this injection. And this is how you're going to inject into the fertilized eggs, where you're going to inject your gene of interest. And what he is showing over here is you're going to flip this part over there. The pronucleus is present over here. They are going to flip it so that it comes down. And once it is coming down, you're going to hold it with a proper needle or a forceps over there, and then you're going to inject the gene of your interest. So this is how you're going to do this experiment of micro injections. So that was about micro injection. Now we'll talk about a method of transgenic fish production by electroporation. This is one more very important technique, and this is also gaining a lot of interest among this biotechnologists over there. So what is this particular electroporation? So this is a method of transformation via direct gene transfer. You don't have to wait for fertilization, or you're not going to wait for microinjection. So this is a direct gene transfer. So this is a method for transferring foreign DNA into bacteria, yeast, plants, and animal cell in the cultures. This technique has become popular for transferring transgene in a fish embryo in the last 3 years. So this is just a budding type of an experiment. So this uses a short electrical pulse to permeate cell membrane, thereby permitting the entry of gene or drugs into the embryo. And this technique, mixture containing cells and a DNA, is exposed to a very high voltage electrical pulses, that is 4,000 to 8,000 volts per centimeter, for a very brief time, maybe few milliseconds. You're not going to let it go on for minutes together. So if you're going to do that, the cells are going to die. So it's just a micro or a millisecond fraction. You're going to just switch it on and switch it off in very seconds over there. So what is happening is, when you're giving this electric current to this particular cells, it results in the formation of a transient pores in the plasma membrane, through which the DNA seems to enter inside the cell and then the nucleus. So what is this? You can see in this particular picture over there. So you have a egg over there, the oocyte over there. What you're going to do is, you have the genes of your interest present outside. You're going to introduce it outside the cells. And then what you're going to do is, you're going to give a signal, electric current is passed to them. Once the electric current is passed, the cell membrane is going to open up very little. You can open it up. And once it is opened for that millisecond, whatever the drugs or the genes that are present outside will immediately rush inside the cell. And once the millisecond is over, immediately the cells are going to close off. So once it is closed off, what is the final result you will find is, there is some amount of drugs or the genes that have entered into this oocyte over there. So that is electroporation cell process, where you're going to transfer the gene directly. That is electroporation. So this is again how this is done. So this is a machine where you're going to give that electric current over there. Now this is a gene of your interest with a beta actin promoter gene over there, and this is a gene of interest over there, and a poly A tail where it is going to stop. Then you have a tilapia brooder over here, tilapia fish. That is what we are talking about. So you're going to collect the eggs and the sperms. You're going to fertilize them over there inside after that spawning. What you're going to do is, after that spawning, the eggs are collected over here, and you're going to add into this cuvette over there. This cuvette is going to be attached to this current over there, and once you give that signals, they are going to get introduced. The gene of interest is going to get introduced into this oocytes. So what is this exact cuvette over here? This is the diagram that is been shown over here. So this is the current that you are going to supply over there, and you have this cuvette over there. This cuvette is something like that. You can see the ready-made cuvettes are present over there. And what you're going to do is, into this cuvette goes your oocytes and DNA of your interest over there. And then what you're going to do is, you're going to supply the current, And once you supply the current to them, the electric electrodes is going to pass the electric shock into this eggs over there. And once the egg plasma membrane is open, the cell suspension, which is having the DNA of your interest, is going to enter into the oocyte directly. So this is how the electroporation techniques can occur. So this is the cuvette wherein you're going to add your cell suspension, and you're going to introduce it into this electrode over there. You're going to place it into this electrode, and you're going to give that electric shock. So that is electroporation technique. So although the overall rate of the DNA integration in transgenic fish produced by electroporation may be equal to that of microinjection or slightly higher than that of the microinjection, but the actual amount of the time required for handling and the number of embryos by electroporation in order of magnitude is much less than the time required for microinjection. So what he's saying is this particular technique is much rapid, and you can do n number of eggs very, very fast, because in microinjection, you have to hold one egg and you're going to introduce it. But in this method, you are going to complete this few number of eggs in a very limited time period. So that is what it says. And electroporation is considered as an efficient and versatile massive gene transfer technology. And as we talked, this is a budding one. Just 3 years back, this has been started over there. Okay. Now, what is the next one? Next one, we are going to talk about gene transfer with the help of pantropic retroviral vectors. So that's a pantropic retroviral vector. So this is one more retroviral vector that you're going to talk about. Remember, we were talking about 4 or 5 different types of vectors. So here we are going to talk about retroviral vector. And although these transgenes can be reproducibly introduced into various fish species, the microinjection and electroporation technique, but the resulting P1 transgenic individual possesses mosaic germlines as a result of delayed transgene integration. Remember, understand what he's saying over here. So when we're talking about the microinjection or electroporation, sometimes what happens due to a little bit delay in this particular work that is being done, what is the product that you get? You get a mosaic germline, that is chimeric germline. Further, these two gene transfer methods are not effective or successful in producing transgene in marine fish and invertebrates. Though you can do it in a freshwater, this particular micro-injection and electroporation is not very successful in marine fishes, that is saltwater fishes and invertebrates. So what is the next step that you can do? This is the new technique, that is new gene transfer of vector with a pantropic retroviral vector. That is a defective pantropic retroviral vector has been developed by Burns et al. In 1993. The vector contains a long terminal repeat. So this is a long terminal repeats sequences on either side of the retroviral genome over there of Molony-Murine leukemia, that is MML virus, Molony-Murine leukemia virus, MOMLV. That's the name of this particular retrovirus. And the transgene packed in a vial envelope with the gene protein of vesicular stomatocytis virus. So remember the gag pollen envelope sequence that you found in the retrovirus over there. So on the same line, so this transgene is going to be packed up in an envelope over there with a gene protein of vesicular stomatocytis virus. So this is packed up and it's going to be introduced into the VSV virus, and this is going to be infecting, or you're going to have a recombinant retrovirus. This gene is ready for transgene into another animal of your interest, whether it can be a fish or whether it can be a cow or a mice over there. So because the entry of the VSV into the cell is mediated by the interaction of VSV-G proteins with a phospholipid component of the cell, the pseudotype retroviral has a broad host range and is able to transfer transgenes into many different types of cell using the pantropic pseudotype defective retrovirus. So you need a pseudo defective retrovirus so that you can transfer the gene of interest packed by VSV gene proteins over there. So this is done successfully in zebrafish. And this is a valuable gene transfer technique for fishes because introduction of the viral sequences into food fish may not be accepted by the public, but it could be used for ornamental fish development. See what he says again, because you're using retroviral viruses, This technique may not be accepted for fish. Food fish means the fishes that we are going to consume. We may not be using this particular process in the food fishes, but nevertheless, this process can be done for ornamental fish development. What is ornamental fish development? You're going to produce some fishes with bright colors, or you can also produce fishes that can glow in the night. Once in the water, they are going to glow. So these are called ornamental fishes, which can be kept in your aquariums of your interest. So this is a pseudovirus. This is the VSVG protein and VSVP protein present inside, and a lipid bilayer, and a firefly luciferase is the luciferase is a glowing type of a gene, where once this is incorporated into the fish is going to glow. Luciferase glowing gene that is present over there. Okay, so transgenic fish have been produced that could benefit the aquaculture industries. So you're going to talk about what are the benefits this aquaculture industry is going to get from this transgenic fishes. What are they? So they enhance the traits of the commercially important species to intensify the growth and the efficiency of the food conservation, to develop fish as bioreactors to produce biomedically important proteins. Bioreactors, what do you mean by bioreactors? Yeah, you can also call them as biofactories, that is live factories for development or production of important proteins. To enhance the utility of fish as indicators to adverse health effects associated with exposure to toxins in the aquatic environment. There we talked about the glow fishes, where Medaka fish, where you introduce them, and if there's any pollution in the water, they are going to glow. And to develop non-mammalian animal models for comparative biomedical research and for functional genomic studies. So a number of fish species are in focus for gene transfer experiment and can be divided into two main groups. Animal uses, it's used in aquaculture, and model fish used in basic research. The major food fish species are katla, labiorohita, tilapia, and salmon. And also the catfish, the zebrafish, the medaka, and the goldfish are used in basic research. So they are used in basic research. Why? Because they're all tiny fishes. They are not very big ones when you're talking about food fishes. Food fishes, when you're talking about the katla, labiorohita, tilapia, and the salmons, because they are going to grow really good enough, and their weight is also good enough for using in the food. But when you're talking about basic research, you are going to use the zebra, medaka, or the goldfish for basic research purpose, because they are going to be small and you have lot number of eggs ready for display purpose. Okay, so these are the constructions of the transgene that you can use. For example, transgene constructions, how you're going to construct when you're going to have a promoter cloning sequence, structural gene cloning, and fusion gene preparation. So that is construction of a transgene. Transgene delivery into the fish, gamete or the zygote preparation is done, and then you're going to do it by micro injection or electroporation technique. Then what you're going to do is found a transgenic fish. That is, you're going to find which fish has taken up that particular gene of interest. So you're going to do some analysis. That analysis is transient expression assay or PCR screening to find out which fish has got that particular gene of interest. And then transgenic progeny F1, how you're going to analyze this? Then you're going to do this crossing of the breeding with back cross and the test cross. Then stable transgene germline, that is analysis of the transgenic loci, phenotype evolution. So phenotypically also you can see which the expression of them, and the Mendelian inheritance of F2 and F3, and homozygous breeding. So when you're doing that homozygous breeding, you will completely know whether that particular gene of interest is being expressed or not. And desired transgenic stock is definitely ready for usage of this. So this is how the step of preparation of transgenic construct is being obtained. Okay, biological tools for disease diagnosis in aquaculture. This is again a very important topic for examination point of view, because at least definitely one major question or two minor questions can be asked in this particular topic over there. So what is that we are going to talk about over here? Why at all you need to know, or you need to have this diagnosis, is because when you're going in a population wherein the most of the population are going to survive only in this aquaculture, that is principal source of livelihood for a large scale section of economically underprivileged population. More than 14.5 million people depend on fishery activities. So these people, they survive only, their job is only aquaculture. So when they're depending so much on this particular aquaculture, so we have to have some biological tools ready to diagnose what type of disease are going to be affecting, or what type of disease the fish has been affected. All this has to be done. Now, when there is no proper diagnosis tool, what happens is sometimes 10 to 15% of the production is lost due to disease problem. And sometimes total loss of $1 billion was reported due to disease in the shrimp culture. Shrimp is a small prawn cultures. Hence, it is essential to take care of this pond and hatcheries to prevent the disease spread. And effective treatment and control of the fish disease requires an accurate diagnosis. And in simplest term, diagnosis of disease is the ability to distinguish one disease from another disease. So you cannot have one particular kit and say that you will be able to find out all different types of disease in what particular. So you have to have different types of diagnosis kit for different diseases. So what are they? So under that, we'll be talking about the first one is ELISA technique, or enzyme immunoassay. So ELISA is called as enzyme linked immunosorbent assay. So enzyme is linked to an immunoglobulin or antibodies, that is called as enzyme linked immunosorbent assay, the full form ELISA. This is also another name referred to as enzyme immunoassay. So you're going to just say enzyme, you're not going to talk about link, but it is just enzyme immunoassay. Nevertheless, both are the same, and these are commonly used to measure antibodies, antigens, proteins and glycoproteins in the biological sample. So you're going to check whether either you're going to check the amount of antibodies that is present in the sera, or you're going to check the amount of antigen that has been affecting the particular fish, or what is the type of a protein, whether it's a toxic protein or a non-toxic protein, and also the glycoproteins in the biological samples. So ELISA is a distinguished analysis compared to other antibody assays, as it yields quantitative results and separation of non-specific and specific interaction that takes place through serial binding to solid surfaces, which is normally a polystyrene multi-well plate. So this is a multi-well plate which is made up of polystyrene plastic. You can see the well over there, the very small wells, wherein you can add almost 500 microliters, 200 to 500 microliters capacity it can hold over there. Very, very small amount. So that's a multi-well, 96 multi-well plate usually they use for this ELISA technique. The antibodies are blood proteins produced in response to specific antigen, and this helps to examine the presence of antibodies in the body in case of certain infectious diseases. So what are the examples? Some examples of here: diagnosis of HIV infection, then pregnancy test is also done by this particular method, and measurement of cytokines or soluble receptors in cell supernatant or serum is done by this ELISA technique. So what is this technique and how it is done? So we'll talk about the procedures of here. So this ELISA can be classified into 3 types depending upon the different methods used for binding between antigen and antibodies. So you're going to talk about antigen and antibody binding, and this is of 3 types. What are they? Indirect ELISA. When you're talking about indirect ELISA, you're coating antigen to the micro titer wells. So this is a micro titer well. Yeah, we saw this micro titer wells. So you're going to coat them with the antigen, a known antigen. For example, if you're doing some, if you want to find out a jaundice or a typhoid, you're going to first coat that particular antigen, which is going to produce that particular disease of interest in the micro titer. And then you're going to add your serum of interest to find out indirect ELISA. In a sandwich ELISA, antibody is coated on the micro titer well. So you know a particular disease, to that particular disease, you know what type of a antibody. So here you are going to talk about monoclonal antibody. So that is specific that for that particular antigen. So antibody is coated onto this micro well plates over there. So that is sandwich ELISA. And in a competitive ELISA, the microtiter well, which is antigen coated, is filled with the antigen antibody mixture. So here there's a competition, which one is going to go and attach onto that particular antigen of interest. So there are 3 different types. We'll talk about them in detail one by one. So this is a simple ELISA technique, that is direct ELISA technique, wherein you're going to just add the antibodies with a antigen coated plate, and then it is the color is read by spectrophotometer. Simple direct. So here in indirect, you're going to have antigen coated, and then you're going to add primary and secondary antibodies. Sandwich method, where you're going to sandwich it with the primary and the secondary antibodies, and another antibody is added over there. And this is a competitive ELISA. All this can be done. And what is the substrate over there? When you add this enzyme linked antibodies over there, what is happening is this enzyme link is going to attach to the substrate over there. And once the substrate is going to attach to the enzyme, they are going to give that color. So that is what is called as enzyme linked immunosorbent assay. So enzyme is linked to the antibody first, and then this enzyme is going to get attached to the substrate, which is attached to some color development. And once it is going to be attached, the color is going to be developed. If it is not going to attach to the enzyme, then the color is not going to be developed. That is the main technique of ELISA over there. So we'll talk about one by one over there. We'll talk about indirect ELISA technique. Developed in 1978. This is the latest one that has been developed. The first one was competitive ELISA technique, but this is recently developed. Developed in 1978. Most popular type of ELISA in use. Antibody detection is carried out in indirect ELISA. Why antibody detection? Because antigen is coated into the microtiter well. So antibody is detected over there. And when a secondary antibody linked with the enzyme is used, the color is being produced. So in this technique, antigen is coated onto the microtiter well. Serum or a sample is being added, which is containing primary antibody, and is added to the micro well and allowed to react with the coated antigen. Any free primary antibody is washed away with the buffer solutions. And then the antigen is detected by adding an enzyme conjugated secondary antibody. Can you see this primary antibody and secondary antibody over there? The secondary antibody is conjugated with that enzyme. Unbound secondary antibody is then washed away by a specific substrate for enzyme, and then a specific substrate for enzyme is added. Enzyme hydrolyzes that substrate to form a colored product. So you can see that S over there, the colored product over there. The amount of color end product is measured by spectrophotometric plate readers, and this can be done by a 96 well plate. So that is an indirect ELISA method. So you can see in this picture over there, the well is coated with an antigen. That is, you know a particular disease. For example, you're going to talk about a typhoid over there. You're going to add that typhoid antigen onto this well over there first. You're going to coat it. Then you're going to add the antibody. That is, what is this antibody? The patient serum. You're going to collect the patient serum, and you're going to add this patient serum into the specific well where you have coated with this antigen. Now, if the patient is suffering from typhoid, the patient must have already prepared those antibodies in their serum. Now, that antibodies is going to get conjugated to this particular antigen. Once they have been conjugated, you're going to wash it with the buffer. Why? Any unconjugated antibodies are going to be removed from that solution. Then you're going to add a secondary antibody, which has been linked to an enzyme. So this is a secondary antibody. The black one is a secondary antibody. The blue one is a primary antibody. And this secondary antibody is attached to an enzyme linked immunoassay. That is what we're talking about. Now, once you're adding this antibody, secondary antibody is going to get attached to the primary antibody. And after this attachment, you're going to wash it off, because if there's any extra secondary antibody, you're going to remove it off, because all those unbound ones are going to be free, and this will be washed away. And after washing, you're going to add a substrate. The substrate is a one that is going to glow once it is going to get attached to this enzyme. So the substrate is going to be enzyme specific. So that particular substrate, that particular enzyme. So you're going to prepare that enzyme, which is going to have that substrate specificity. So this substrate is going to attach to the enzyme, and this enzyme is going to hydrolyze that substrate, and thereby the color is being formed. The color formation depends on the intensity or the degree of infection that person is going to be affected. So that is indirect ELISA technique. So you can read this particular topic of how the procedure is dealt with. Okay. Now, what are the where all you can apply? You can apply for HIV, rubella, dengue viruses, etc., and to detect certain drugs in the serum. So you want to detect any drugs, excess drugs present in the serum, you can also do this indirect ELISA technique. What are the advantages? Increased sensitivity. A wide variety of labeled secondary antibodies are available commercially, so very easy to do. Maximum immunoreactivity of the primary antibody is retained, and then the versatile, because many primary antibodies can be made in one species, and you can also prepare the secondary antibody for that particular primary antibody. Flexibility, since different primary detection antibodies can be used. Cost saving, since fewer labeled antibodies are required, and different visualization markers can be used. That is the substrate that we are talking about. Different visualization marker can be used for the same primary antibody. Only disadvantage is cross-reactivity might occur with a secondary antibody, resulting in a non-specific color production, and extra incubation step is required in this particular procedure. So that was about indirect ELISA application. Now, talking about sandwich ELISA over here. What is this? Developed in 1997. Indirect was developed in 1978. Sandwich was done before a year before, that is in 1977, for detection of antigen. There it was detection of antibodies. Here in sandwich ELISA technique, you're going to detect the antigen with the help of the antibodies. So here you need two types of antibodies. First one is a capture antibody. Capture antibody is ready to capture the antigens. And the second one is enzyme-linked secondary antibodies. This is going to get attached to the primary antibody, capture antibody. So that particular secondary antibody is going to have an enzyme link. So the antigen interacts first with the capture antibodies, and then the secondary antibody binds to the epitope of the capture antibodies. And when you're going to add the substrate, the color is being produced. So in this technique, antibody is coated onto the microtiter well. A sample containing this particular serum is added to the well and allowed to react with the antibody attached to the walls of the microtiter. After the well is washed, the second enzyme-linked immunosorbent assay is added so that it gets bounded to the capture antibody, and any unbound ones are washed off with a buffer. And finally, you're going to add the substrate, wherein the enzyme is going to hydrolyze that particular substrate, and it is going to give that color. So mainly used for detection of rotavirus and enterotoxins of fecal E. coli, and pregnancy test kits is also based on this sandwich ELISA technique over there. So what is the sandwich ELISA technique? So here you're going to have the microplate. The microplate is coated with the monoclonal antibody, particularly of that particular interest. So here we are showing only one well over here. Remember, we are doing this in a 96 well plates. Okay. So this is only one well we are showing over here. So this is coated with a monoclonal antibody, and then you're going to add the serum, the patient serum over there. So any antigens that is present into this patient serum over here in this particular plate over here is going to get bound onto this capture antibody. So this is a capture antibody. So these red ones are the antigen that have been captured by this antibody. Then any unbound antigens are going to be washed off by the buffer over there. And then after washing, you're going to add a secondary antibody, that is a monoclonal antibody that has been linked to an enzyme over here. So any antibody which has an E attached over there, that means you have to say that is enzyme linked antibody. So this enzyme linked is always a secondary antibody over there. So what it is going to do is it is going to get attached, the epitope is going to get attached onto the paratope of this antibody. Remember immunoglobulins and how they are, where you have the epitopes and the paratopes over there. Epitopes are found on the antigen surface and paratopes are found on the antibody surface. So here what is happening is this is going to get attached onto this antigen, and some sort of a sandwich type is occur over here. Sandwich, you have two breads, and in between whatever the layer that you have, you're going to keep it. So that is called a sandwich technique. So here the antigen is being sandwiched by two antibodies. One is primary and one is the secondary over there. So the name sandwich ELISA technique. Now, once this secondary antibody has been added, it is again washed. Why? Because any unbound secondary antibody is washed off. And then you're going to add the substrate of that interest. And once you add the substrate is going to be hydrolyzed by the enzyme and the color is going to be developed. And the intensity of the color is read by the spectrophotometer. So what is the advantages of here? High specific, since two antibodies are used to capture the antigen, suitable for complex samples since the antigen does not require purification, and flexibility and sensitivity is done in both direct and indirect detection method. So that is sandwich ELISA method. Now coming to competitive ELISA technique. As I told you, competitive ELISA technique was done in 1976. This is the older among all the other sandwich and the indirect ELISA technique. So this is done for detection of antibodies. Your competition, what is a competition over here? The principle of this test is that two specific antibodies, one conjugated with the enzyme and the other is present in the test serum itself. And these are used for this particular technique over there. So there's a competition that is occurring between these two antibodies for the same antigen. The appearance of the color indicates negative test, that is absence of antibodies. And the absence of the color indicates positive test, presence of antibodies. Now, what is this negative test and positive test colors technique that is present in this competitive ELISA technique? Now, when you are seeing the procedure, you will understand this. But remember that appearance of the color indicates negative test because there is absence of antibodies. When do you have antibodies? When the person is affected with that antigen, your body is going to produce antibodies. Clear? Now, when there is absence of antibodies means there is no antigens present. So there's a color development occurring over there. That gives you a negative test. But if it is positive, that is, there is a presence of antibodies. These antibodies are going to get attached to the antigens and there is no color that is being produced. So that technique is that is going to give you a positive test because there is absence of color. So remember, presence of color negative.
Can you explain once again?
Yeah, I'm going to talk about that procedure in the next slide. You will understand this. What I want you all to understand first is when you have the color indicates negative, when you have no color indicates positive test. So what is that? We'll be talking about this in the particular next slide over here. So in this test, antibody is first incubated in a solution with a sample containing the antigens present in that serum. So you're going to take the antibodies and you're going to take the test serum and you're going to incubate them for some time, right? Then what you're going to do is you're going to add it to the well. So let me put it over here. So this particular experiment is done for detection of HIV. So we'll talk about this particular experiment over here. Now, what is this present over here? So antibody is incubated with a sample containing antigen. So for example, there's a person who is suffering from HIV. Okay, so what you're going to do is you're going to take that serum from that patient sample and you're going to incubate with that antibody specific to that HIV antigens. Followed? So you're going to incubate it first outside. You're not going to directly put it into the plate, micro well plates over here. So first outside, you're going to add that antigen and antibody. What is the antigen? Patient serum is an antigen because you think that he's been infected with HIV. And you're going to add it into this monoclonal antibody. So you have incubated that first. Then what you're going to do is you're going to add that into a micro, the micro titer plates, which is already coated with the antigen. Which antigen? That HIV antigen. Followed? So this antigen antibody complex are added to a well, which is pre-coated with that particular antigen. So these are the antigens over there. Okay, so what is happening now? Once this is added, then you're going to add it and incubate it for some time, and you're going to wash the plate to remove any unbound antibody. So for example, over here, you already have, for example, you have added antibody and you have also added antigens over there. And then what happens is, if the patient is suffering from HIV, the antibodies that you have added into that solution, it's going to get bound to the antigens of that particular patient. And when you're going to add the solution into a pre-coated well, which is already having the antigens, any unbound antibodies that is present is going to get attached over there. And if there are more number of antigens, more number of antibodies going to get attached to the patient's antigen. So there is no more free antibodies present. You're going to wash it. And then what is the next step? You're going to add an enzyme-linked immunosorbent antibody over there. That is a secondary antibody. And then what you're going to do, you're going to add, that is the fourth point is enzyme-linked secondary antibody, which is specific to that primary antibody is added. Wash the plate so that the unbound enzyme-linked antibodies are removed. Add the substrate, which is converted by the enzyme into fluorescence over there. So you have a fluorescence signal. The higher the concentration of the antigen that is being present over there, the lower the absorbance. Higher the concentration of the antigen, lower the absorbance. So remember over here, where is that point over here? So here, if you're having more amount of antigens present, the antibodies that you have added to incubated is already being bound to them. Now, once you are going to add that solution into an antigen plated well over there, if you have no antibodies, they are not going to get attached over there. If you have more antibodies, all those antibodies are going to get attached onto this antigen plate over there, and those that are bound are thrown out. Now, once you add a substrate, if you have more antibodies present onto this plate, it is going to glow higher. The maximum color is expressed. When there is a maximum color that is expressed, that means the patient is not suffering from any virus. If the color is less, then you are going to say that that patient is suffering from that particular virus, because all the antibodies had been joined to these antigens that is present in the serum patient sample, and there is no more antibodies that is going to attach onto this plate of the antigen so that the substrate can give that color. So what he says over here, this is one more perfect example over here. So here the positive samples over here, and this is a negative sample over here. Positive sample is all the antibodies that is added, they are going to be bound to the patient's antigens, and there is no more left over here to attach to this antigen that has been coated onto this plate. So they are being washed off. Once all the antibodies are washed off, there is no more enzyme linked immunosorbent to attach to the substrate of that color, to give that color. Okay, so they're all washed off and there is no color formation over here. So this is positive result. But if the patient is negative for that virus, in his sample, there will be no antigen present. And when you're adding that sample with the antibodies, there is no antigen being present. The antibodies is not binding to any antigen in the sample. But when you add the sample into this plate, which is coated with this antigen, the free antibodies are going to get connected to this particular antigen. And when you're going to add a secondary antibody, which is enzyme-linked immunosorbent like that, and we're going to add an substrate, this particular sample is going to glow. Once if it is going to show the color, that means the patient is not suffering from any virus. So he is not having any antigens. So that is competitive ELISA technique. Follow this point? Any doubt of that?
No, ma'am.
Understood this point now?
Understood, ma'am.
Right.
Yes,
So
ma'am.
competition is competition for the antigen antibody that has been occurring over there. So that is what is called competitive ELISA technique. So what is advantages over there? They are highly specific since two antibodies are used. Highly sensitive since both direct and indirect detection method is can be used over here, and suitable for complex samples because antigen does not require purifications over there. So that is what is competitive ELISA technique. So they might ask you to talk about the diagnosis technique, disease diagnostic techniques over there. So when they ask you this question for major question, you are going to talk about ELISA, indirect ELISA technique, sandwich technique, and competitive technique. In case they're going to ask you for a short answer, then they might ask you to write in about any one or two diagnostic technique, that is either ELISA or competitive. So you can draw this diagram so that the examiner will understand that you have understood what you're writing in that particular answer there. Okay, so applications of ELISA, the whole ELISA technique, where all you can apply them. Presence of antigen or presence of antibody in the sample can be evaluated. Determination of the serum antibody concentration in a virus test. And these are also used in food industry where detecting potential food allergens. Pregnancy kit is also done on this ELISA technique over there. Qualitative and quantitative estimations of various proteins, hormones, and toxins can also be done. Applied in disease outbreaks during the spread of disease like HIV or cold or cholera or STD, etc. And this was done during COVID-19 breakout. ELISA technique was carried out during COVID-19 breakout. So because this was a rapid testing kit, something like your pregnancy kit that you had, you're just going to add your sample and you're going to check for the double lines over there for color. If you have a double line, then it is positive, and if you have a single line, then it is negative. So that this ELISA was also done during this COVID-19 outbreak over there. Okay, so coming to the next one, polymerase chain reaction technique. This polymerase chain reaction technique is going to take another half an hour or so. So I think we are running short of time. We will continue this topic in the next class, because for now, you understand what is this ELISA technique and what is this transgenic techniques that we did, that is gain of function, loss of function, and the retroviral transfers that we have done. Then we talked about micro injection in the eggs, and also we talked about electroporation. Sometimes you might also be asked in a short answer to describe about electroporation. So there you're going to discuss about this electroporation, and the diagnosis also that was done, ELISA technique. Another diagnosis we are going to do is polymerase chain reaction, and this polymerase chain reaction we will continue in the next class. Okay, so any doubts, Ma, regarding this topic that we did till now?
No, ma'am.
No. Okay, so no, ma'am. Fine, very good. So in case you still have any doubts, you can ask me. And one more point I wanted to ask is, how do I send this PPTs for y'all? Because I'm not an admin over there. So okay, let me, if you have any group, if you can send me the link, then I'll post all these PPTs for y'all over there. Fine?
Yes, ma'am. Okay, ma'am.
Okay, yeah. So see y'all in the next meeting.
Thank you, ma'am.
Good evening,
Okay.
ma'am.
Good evening.
Good evening, ma'am. Thank you.
You.